Good morning to the lecturers and my fellow batchmates. I am Chung Kenyan. Today, me, Xiang Chen, and Pui San will be presenting a topic called Gardia lamblia. So, actually, what is Gardia lamblia? Gardia lamblia is an isolated intestinal protozoa in the world, and in fact, is the most frequent intestinal protozoa in the world. Around 200 million cases per year are happening in the world because of intestinal protozoa. This intestinal protozoa is found on surface of soil or in soil, food, or water contaminated with feces from infected human or infected animals. The prevalence because of intestinal protozoa is higher in poor sanitation area, like for example, developing country like Africa and India. The rate of infection is the highest among children in daycare center and among patients in mental hospital because of poor hygiene possibility. And sometimes it may cause outbreak because of pure water hygiene management. So Gardia lamblia can actually exist in two forms. One is trophozoite and another one is cyst. Usually, cysts are present in the stool of infected persons. So, from here, we can see that Gardia lamblia is actually can be transmitted through direct fecal oral route, ingestion of water and food contaminated with feces containing cysts, or maybe oral anal contact, especially in male homosexuals. So now, I would like to pass to Xiang Quan.
Hello everyone, my name is Chen Xiangquan, and today I'm going to explain the life cycle of Gardia lamblia. Gardia species have two major stages in life cycle. The first stage is trophozoite, which is active form, and the second stage is cyst, which is the inactive form. Trophozoite is the replicative form of Gardia lamblia. They are in the disease-causing stage and they can only survive in the small intestine of the host. On the other hand, cyst is the non-replicative form of Gardia lamblia. They are in the transmission stage. Moreover, cysts can survive for prolonged period outside of host in most ambient conditions. The life cycle of Gardia lamblia can be complete in one host. The infection of a host is initiated when the cyst is ingested with contaminated water or food through fecal oral contact. After exposure to the acidic environment of the stomach, cysts excise into a trophozoite in the proximal small intestine. Trophozoites swim through the intestinal mucus until they eventually adhere to the host intestinal epithelium. Then they divide by binary fission, forming either more trophozoites or the non-replicative cyst stage and cause symptoms of diarrhea and malabsorption. After that, the cyst and trophozoite pass through the host's large intestine and are shed in the feces. The cycle repeats and a new host is infected.
Clinical features of Gardia lamblia. The incubation period for Gardia lamblia is around 1 to 14 days, which differs from one another. The onset is sudden, which consists of foul-smelling watery diarrhea. It may not develop any signs and symptoms, where approximately 50% of infected human is asymptomatic. If the symptoms occurs, the symptoms might develop around 1 to 3 weeks after the exposure. Moving on, the sign and symptom. The most significant sign and symptom is watery, foul-smelling diarrhea, also known as soft and greasy stool. The patient might also experience fatigue, malaise, abdominal cramps, and bloating. These signs and symptoms may last for around 2 to 6 weeks or longer, and it may reoccur. Moving on to the laboratory diagnosis of Gardia lamblia. we are going to introduce 4 laboratory diagnostics which are commonly used, such as ova and parasite examination, fecal immunoassay, strain test, also known as enteral test, and polymerase chain reaction (PCR). Ova and parasite examination. Ova and parasite examination is used to determine is there any parasite infecting the digestive tract by using microscopic evaluation of the stool sample. For the morphology of Gardia lamblia, the cyst is in oval shape with 4 nuclei, while for the trophozoites, is in pear shape with 2 nuclei and 4 pairs of flagella. The next laboratory diagnosis is fecal immunoassay. It is a more sensitive and specific test to test for Gardia lamblia. An ELISA test that detects a Gardia cyst4 antigen in the stool can be used. ELISA can provide a useful measurement of antigen or antibody concentration. For the string test, it is carried out by swallowing a wet piece of string until it reaches the duodenum. In the duodenum, the string absorbs a small amount of duodenal fluid. The trophozoite adhere to the string and can be visualized after withdrawal of the string under the microscope. The last laboratory diagnosis is polymerase chain reaction (PCR). In this case, PCR is used to identify the subtypes of Gardia lamblia and for the specific detection of Gardia lamblia DNA in stool samples. That's all from me. Thank you.